record. Okay. Oh, I know what's going on. Just a second. There we go. Okay, so, all right, let's get started. Okay, so we left off on Tuesday talking about, talking about protein synthesis, right? So what we're trying to do is we're trying to figure out how we can go from genotype, so the nucleotide sequence that we have in our DNA, and how we turn that into a phenotype. And remember, phenotype is the physical manifestation of that nucleic acid sequence. And so what we have to do is take that code, take those nucleotides, and somehow make that into a physical product. And what we're talking about, the vast majority of these products are going to be in the form of protein. So that's what we're going to be talking about today, is how we go from nucleotide sequence to physical protein. So we already went through the first part of this, so just a reminder that when we do this, we go from nucleotide sequence of DNA, our genome, and then we make a copy of that nucleotide sequence in the form of RNA. Specifically, this is mRNA, or messenger RNA. And then what we're going to talk about today is how we take that mRNA and read it into protein. So just a reminder that protein synthesis, or gene expression, or central dogma, all of which mean the exact same thing. What we're talking about is going from genotype to phenotype. So again, nucleotide sequence in the DNA, making a copy of that DNA in the form of RNA, which is the process of transcription, of which there are 3 steps, which we will review. And then taking that mRNA and turning it into protein, which is the process of translation, which we're talking about today. It's just worth noting that here, not D, but we did talk about something that happens between transcription and translation, which is post-transcriptional modification. And we will review that. But just putting that in here, that happens right here, and that's basically capping, adding a tail, plus splicing. Right. So that happens between transcription and translation. So we'll review that real quick. Just a reminder that we are not transcribing and translating the entire genome all at once. We're doing this just for individual genes at a time, segments of the DNA. This is just going through transcription. Remember, for transcription, what we're doing is taking DNA and turning it into RNA. We start at the beginning of a gene, which is the promoter, and we transcribe that segment all the way to the end of the gene, which is the terminator. How we initiate this process is we have our functional enzyme, which is RNA polymerase, start at the promoter, and it reads all the way to the terminator. And what it reads is it reads complementary nucleotides. So remember, very importantly for transcription, what we're using as the template for our mRNA strand is the 3 to 5 prime strand of DNA. This serves as a template that we're going to use to build our complementary strand of mRNA. And remember, mRNA is always, 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 always in the 5 to 3 prime direction. So you can see that's why we have to use the 3 to 5 prime strand. What we do is RNA polymerase opens up the DNA just a little bit, and our RNA polymerase starts adding complementary nucleotides. So if our DNA strand, which is shown in blue, has a sequence CTGAC, then our complementary RNA would be, if we have a C, a G, if we have a T, an A, G, C. If we have an A, because this is RNA, we wouldn't have a T, we would have a U, because RNA has U's, and CG, so on and so forth. all the way from the promoter to the terminator. Once we reach the terminator, it basically just disassembles. And then what happens is that post-translational modification, which is your mRNA processing, all we do is we add a 5' cap, which is just some modified guanines stuck on the end of the 5' end, a bunch of adenines popped onto the 3' end called a tail. And basically what these do is they function as sort of like a zip code, telling the mRNA that it needs to leave the nucleus. If there is a nucleus, it needs to leave the nucleus, and telling it what ribosome to associate with, a free ribosome or a bound ribosome. Importantly, neither the cap or the tail is actually translated. So it does not contribute to the end protein. It's just a protective structure and sort of like a guiding structure. And then another thing that we do as part of the mRNA processing, sorry, I can't believe it, but I'm chilly. Another thing that we do as part of the mRNA processing is we splice out intronic segments of that mRNA. So just a reminder that in DNA, and you can see this in the blue, in DNA, there are both coding and non-coding sequences. The sequences of DNA that contribute directly to protein or the product are going to be the exons or the exonic segments. But there are these little spacers in here called introns or intronic segments, and those have to be cut out because they don't contribute to the protein. So that's called splicing, where you basically just remove introns specifically from mRNA. So that's a really important distinction. Introns and exons are present in DNA. After transcription, you have introns and exons also in mRNA, and we remove those introns from the mRNA. So they're never removed from DNA, but they are removed from the mRNA. So after that processing, after we have the cap and the tail, after we've been spliced, then that mRNA can, if this is a eukaryotic organism, it can leave the nucleus, enter the cytoplasm, and associate with a ribosome. So, cool stuff. I had a bunch of summaries for that. So now, kind of like reviewing just where we started on Tuesday talking about translation. This is just a summary of transcription. Again, what we did is we took DNA and turned it into RNA. We have processed our RNA, and now what we need to do is translate our RNA into protein. So just a reminder that mRNA, whenever we talk about turning mRNA into protein, it's important to know the structure of a protein. So I'm kind of jumping ahead here, and we'll back up again to review what we talked about. But it's important to know that proteins are made out of amino acids. So these are your amino acids. We have we have not a ton of amino acids. We have like 20 amino acids. And they are shown in this table. So phenylalanine, leucine, isoleucine, valine, alanine, proline. These are all amino acids. So we're not going to get into the difference between essential and non-essential, but I can tell you just briefly that the essential amino acids are those that you have to get through the diet. You have to take them in through dietary sources. Whereas non-essential amino acids, the reason they're non-essential is because your body can make them. So it's not essential that you get them through the diet. Complete proteins, which would be something like a plant complete protein would be quinoa, but a majority of proteins are going to be like meat sources because well, muscle is made of has a lot of protein, and proteins are made of all your amino acids. But there are plant-based complete proteins. But again, this is just showing, this table is just showing kind of all of the amino acids that exist. It's not breaking them into essential versus non-essential. But the reason that's important is because you have to have amino acids to build protein. And we have to be able to, what the RNA does is it basically tells us the sequence of amino acids that will be used to make a protein. So just a reminder of why this is so important, the amino acids that contribute to a protein, is really, really, really long time ago, or it seems that way now. Not long enough, in my opinion, but it seems like a long time since I've seen you. In class, we talked about homeostasis, and we talked about the importance of maintaining homeostasis. And the reason homeostasis is so important is because homeostasis is that set of conditions that allows your body's structures or an organism's structures to maintain that structure, right? Homeostasis is where your proteins are in the correct structure, essentially. Well, all your structures, but we'll talk about proteins. If you get outside of homeostasis, so if it's too hot or too cold, or if you have fluctuations in acidity or anything like that, what happens is you can disrupt the structure, and that would, remember, because structure equals function, that would disrupt function. So that would be denaturation. So that's just a reiteration of something that, again, is one of the most foundational concepts in biology, that structure equals function. What we're looking at here on this picture is the most primary structure for proteins. Proteins are made out of amino acids. And at their primary structure, proteins are basically just amino acids linked by peptide bonds. So the reason our mRNA sequence, right, the reason our mRNA sequence is so vital is because it's the sequence of mRNA, the sequence of these codons, that tells us the sequence of amino acids in a protein. So it's the sequence of codons in mRNA that gives us our primary protein structure. So that's why it's so, so, so important, right? It's extremely important because this is where we get that foundational primary structure. And again, structure equals function. So if we look at our mRNA, again, when we talked about going from DNA to RNA, you're doing that at the complementary nucleotide level. But when we talk about going from RNA to protein, we're kind of switching languages. Instead of going nucleotide to nucleotide, we're going nucleotide to amino acid. So in order to translate that, we convert that nucleotide language into a codon language. So basically every 3 nucleotides is a codon. And if we look at that, the codon table tells us what sequence of nucleotides codes for what amino acid. So we briefly mentioned this on Tuesday, but I want to reiterate it here, that this language, our the genetic code, wow, the genetic code is referred to in a couple different ways. I have always heard of it as repetitive. And the reason it's repetitive is you can see that if we need a phenylalanine in a protein, you can code for phenylalanine with 2 different 3 nucleotide sequences. 3 uracils would code for phenylalanine. 2 uracils and a cytosine would also code for phenylalanine. So it's repetitive. You can see leucine is very present in a lot of protein. It has 6 codons that code for leucine. You may also hear the term degenerate for the codon table. Some textbooks call the fact that there is this repetitiveness degenerate. I don't know if that's like a difference between ecologists and medical or human biology related fields, because I had never heard that. But then I talked to Professor Chichi, who teaches 182, and she's like, oh yeah, it's degenerate. I was like, what? Okay, so so yeah, so you may hear repetitive or you may hear degenerate, but basically both of those things are just referring to the fact that multiple codons will code for an amino acid. Another thing I want to reiterate about this table is that when we talk about translation, there has to be a start and an end, right? Where do we start? Where does our complex start for translation, and where does translation end? So translation always starts at the first sequence of nucleotides that is adenine, uracil, guanine, or AUG. AUG, that codon codes for the amino acid methionine, or MET. It's important to note this will not be the only MET in a protein sequence or peptide sequence, but the very first methionine or the very first AUG sequence is where translation will start. And then translation will go all the way down our mRNA sequence, translating these codons into amino acids. And this will continue until that we reach one of three sequences in the mRNA, UAA, UAG, or UGA, which are called stop codons. So we'll talk about what happens at each of those. But translation starts at the start codon, which is always AUG, the first AUG. And translation continues until it reaches a stop codon, of which we have three, UAA, UAG, UGA. To answer the question that I get a lot, which is, do you need to memorize this? Do you need to memorize your stop codons? Absolutely not. I would love you to know that your start codon equals AUG, which codes for methionine. I think that that would be an excellent, excellent, excellent thing for you to memorize. All right, so let's talk about how we do this process now. Okay, so it's not that aggressive, and I just felt the need to stretch there. Okay, so we're looking at our mRNA, and what our goal is, so reminder of our goal is to create a protein. So this is our primary protein structure. We have amino acids, and importantly, I just want to point out that these amino acids in proteins are linked by a special type of bond called a peptide bond. This is why sometimes proteins are referred to as polypeptides. Poly means multiple, peptide is referring to the type of bond. So a polypeptide just means that there's multiple peptide bonds in between those amino acids, and that is basically exactly a protein. In some cases, actually I asked for clarification from a molecular biologist about this. In some cases, really, if you want to get hyper specific, if you're going to go into like protein chemistry or something like that, a protein is anything over 50 amino acids, whereas a polypeptide is anything that is less than 50 amino acids long. Do you need to know that? I didn't until I was 30. So all I will tell you is that, and when I was 30, I had been at this for a couple years. So wait a second, I'm 25. No, but this is kind of how you can, like, to get really specific, this is how sometimes it is organized. For all intents and purposes, they are the exact same thing. My husband just walked in with 1 iced coffee, not 2. Let's see how long he makes it through this quarantine. Okay. All right, so again, what we're trying to do is we're trying to convert this mRNA into protein. And in order to do that, we have to take 3 nucleotide sequences or codons that code for those amino acids. And then we just bind those with polypeptides. I just told my students that you're not going to make it because I see only one iced coffee. Get out of here.
You weren't going to be able to have it for the next like 90 minutes.
Excuses. Okay.
Are you on?
Yeah.
Oh, cool.
Okay. Okay, so that's what we're doing here. Okay, so we have our mRNA strand. It's 3 nucleotide sequences coding for codons, and those codons code for amino acids. We're trying to get a strand of amino acids linked by peptide bonds. Okay, so we talked about the components for this process. We need mRNA, which is going to be our template. What brings those amino acids to the ribosome, which contributes to the growing amino acid strand or protein, is going to be a molecule called tRNA, which we talked a little bit, and I'll revisit. And then this entire process occurs at a ribosome, of which we have 2. So we revisited that on Tuesday, but I'm going to move forward now. This is just showing the structure of a tRNA. So this is kind of showing its crystal structure from the side. So that's why it looks a little bit different. I think in the textbook, they referred to this as the clover leaf structure, which I appreciate you bringing up last time. But this is sort of looking at it from the side as opposed to that clover leaf structure. But regardless, you're going to see the same thing, which is tRNA on the bottom have an anticodon, which sort of recognizes codons. And then on the tip, they carry amino acids. Again, you're going to have tRNAs that carry both essential and non-essential amino acids. The only difference is that essential amino acids are those that were consumed through the diet, just kind of grabbed up by tRNA, and non-essential amino acids were created and then the tRNA grabbed them. Okay, so tRNA has an anticodon that just tells us, kind of helps the tRNA recognize the mRNA sequence. And again, we have codons that code for those amino acids. All right. So this is this was my modified version of a tRNA. And this is kind of just showing how this process takes place. So we have our mRNA. You'll note that the first sequence of nucleotides I wrote out here is AUG, which is always going to be your start codon. The very first start codon, if we look at our table, again, we say AUG. We're looking at the codons in our mRNA for this table. So AUG codes for methionine. Whoops. So what we have here, AUG, that means our first tRNA is going to carry methionine. Our second codon, CGA, we can check the table. CGA codes for arginine. And sure enough, our next tRNA is carrying arginine. So we just go codon to codon through the mRNA, and this tells us the sequence for this protein would be methionine, arginine, serine, and asparagine. And then, like we mentioned, those are linked by peptide bonds. Okay, so we already did this last week, so I won't go through that one. Or Tuesday, not last week, sorry. But I want to go ahead and move forward to the process of translation. Okay, so I have mentioned that we need mRNA, and we need tRNA, and we need a ribosome. So remember that we have free and bound ribosomes, but regardless of where a ribosome is located, it has this really important structure. And it is the structure of a ribosome that sort of allows translation to take place appropriately. So I want to talk about ribosome structure. So ribosomes have 2 subunits. they are made of 2 parts, and basically that's important because you have an mRNA strand, and what happens is the ribosome kind of clamps onto it. So it will have your mRNA, and it just clamps on. So you can see once it clamps on, it has 2 subunits. It has the large subunit, okay, that's top basically, and then it has the small subunit, which is basically the bottom. It's the small subunit to which mRNA binds. You can see the mRNA binding site. And then the large subunit, the reason it has these sort of larger tunnels, the large subunit is where your tRNA come in. So we'll look at this process here in a minute. But your small subunit binds to your mRNA, and your large subunit is sort of works with recruiting and organizing your tRNA. So you have 3 binding sites, or 3 sites in that large subunit, I should say. You have the E site, the P site, and the A site. So these all refer to sort of like what happens to the tRNA when it's in the ribosome. So first, and the way this ribosome is kind of going to read is going to be from left to right, right? So this means that tRNA are going to have to come in through the right side of the ribosome, and then they will leave through the left side of the ribosome. And when I say left or right, I'm referring to like this image, right? So that doesn't make a whole lot of sense molecularly. But for this image, what's going to happen is tRNA are going to come in or enter the ribosome on the right in what is called the A site. So you can see the A site is where your tRNA first bind. They first enter the ribosome. And then what happens is the ribosome moves to the right on the mRNA strand. And so if a tRNA, I'll try to like figure out if I can do this actually, just for sake of simplicity, here is my tRNA. There we go. Okay, so here's my tRNA. So what happens is it enters the A site, right? So remember the bottom of the T is what binds to your mRNA. It enters the A site. And then what happens is your ribosome moves to the right. So it moves this way on the mRNA strand. If the ribosome was to move to the right and the T was held in place, what happens is the tRNA would be moved into the P site. So the P site, what that stands for is peptidyl tRNA binding site. What I like to remember that by, what just happened? I like to remember that instead as P for peptide bond. So that kind of tells you what happens in the P site is, remember we have our tRNA enter the A site. When the ribosome shifts over, that T gets moved into the P site. And that's where we're going to have our amino acids linked by peptide bonds. So you see this exit tunnel. It's a little bit misleading. The exit tunnel is where you have your polypeptide strand kind of held in place. So it kind of look like that, right? Kind of coming off that T. All right. So then what would happen, because this shifted over, you would actually have room for your next tRNA. All right, so let's just have that. It's a terrible color choice. You would have room for your next tRNA to enter the A site. Okay, cool. So remember, let's do this process again. You have your first tRNA enter, the ribosome shifts to the right, that moves that tRNA, and then that makes room for this next tRNA to move in. We have our peptide bond built in the P site. So then what happens is the ribosome shifts to the right again, and this moves both of these tRNAs over. So you'll notice that the polypeptide strand didn't move. It was shifted from this T to this T. But now we have this T, it's done. And you'll notice this site is called the E site. That's referring to the exit site. So what happens when then your tRNA enters that site is it's done. It leaves. All right. And then again, this T was shifted into that spot, and then it leaves room for the A site to be open for the next tRNA to enter. So again, we'll look at this process again, but if you just kind of visualize that happening, I can remind you that your very first tRNA is always going to carry the amino acid methionine, because that's your start codon. So what happens, again, is you have your first tRNA enter the A site, your ribosome shifts to the right, which moves that tRNA in there. The methionine is going to be contributed to the polypeptide strand. This left room for this tRNA to enter the A site. And then that means we can move the T. Notice that your amino acid was left. It was attached to that growing polypeptide strand. And then that allows this one carrying its amino acid strand to contribute to that. Shift again. And that tRNA can pop out of there. Okay. So again, we will look at all this. Oh my Lord in heaven. Okay. I apologize. Okay. So that's kind of what happens. And again, we're going to look at this process all take place. So the A site is where tRNAs enter the ribosome. They shift over into the P site. That exit tunnel is where we have that polypeptide. And then when it shifts over again, the E site is when a tRNA that has kind of dumped its amino acid, it's allowed to exit. All right. Any questions so far? We will look at this process on the next couple of slides as well. All right. Any questions about the ribosome or any of the components needed? All right. Okay. So I'll move on. All right. So let's look at this process. Just a reminder that just like transcription, translation has 3 steps: initiation, elongation, and termination. So for translation though, where we talked about transcription, the only thing we needed to do for transcription was have RNA polymerase bind. For translation, there's a little bit more involved with it, but that's just because there's a lot more components involved. So what we have to do for translation, the very first thing that happens, and just to orient you to this picture, you can see that our mRNA is shown in pink, and it is 5:3. So ribosomes, here's a very important thing for you to know. Ribosomes read mRNA in the 5:3 prime direction. So this is really important because remember, we talked about the fact that mRNA really, really needs to be in the, will always be in the 5:3 prime direction. The reason this is so important is because remember, in order to get that protein structure, we have to read everything in sequence, because protein sequence, amino acid sequence, is that primary protein structure, and structure equals function. So we have to have the correct order. In order to have the correct order, we have to have a start and an end, and that has to be directed. Where do we start and where do we end, right? And the sequence depends on that. So ribosomes will always read mRNA from 5:3. So when I mentioned it moves right, realistically what I meant, kind of molecularly speaking, it's moving from the 5' end toward the 3' end of that mRNA. This is just kind of like, this textbook is based on reading the English language. We read left to right, which is why this is showing 5:3 and moving left to right. But in all reality, the directionality for these strands is 5:3. The ribosome moves 5:3. Okay, so we have our mRNA strand, the 5:3 prime strand. what happens, that mRNA left the nucleus, if it's eukaryotic, and the ribosome clamps onto that mRNA. Remember that the mRNA's binding site is on the small ribosomal subunit, which is shown in this kind of like darker orange color. And then our tRNA enter the ribosome and kind of interact with the ribosome at the large ribosomal subunit, which is shown in sort of this like lighter orange color. You can see all three sites, the E site, the P site, and the A site. And you can see what happens is something that is very important. And we talked about the fact that tRNA enter the ribosome complex through the A site, but there's one exception to that rule. Of course, there's always an exception. The one exception to that rule about tRNA entering at the A site is that very first tRNA. So before this, when I was showing that sort of like animation where I was moving stuff, I was showing basically elongation. But because that's how the majority of tRNAs enter. But the one exception to that rule is the very first tRNA that enters the ribosome complex. Remember that tRNA is carrying methionine because your start codon, which is shown in green, your start codon is always AUG, which codes for the amino acid methionine. So that's always going to be your first tRNA. I want you to look at this picture for a second, though. You will notice that towards the 5' end, AUG is not the first 3 nucleotides on that mRNA strand. Okay? You can see here there's at least 2 codons in this picture that come before AUG. But regardless of what codons are present before AUG, it doesn't matter because translation will not start until that start codon, AUG, no matter where it is, right? Okay, so what happens to initiate translation? The first part of initiation is that we have this complex created. We have our large and our small ribosomal subunits. The P site is where you have that first codon located, AUG, and then our very first tRNA enters the P site. This will not happen for any other tRNA subsequently. It is only the first tRNA carrying methionine that enters the P site. All subsequent tRNAs will enter the A site. Okay, so we've basically assembled our complex. We have our ribosome bound to the mRNA. In the P site, we have our start codon, AUG, and we have our first tRNA in the P site carrying methionine. So there's more that happens during initiation for translation. So the next thing that happens during initiation to get this process started is basically like, okay, we assembled the complex, now we kind of just need it to like get moving. So what happens for the next part of initiation is you'll notice we had our complex, our small and large subunits, our mRNA. We had our very first tRNA bound in the P site carrying methionine. Another thing that gets this process started is you have your second tRNA enter the A site, and all subsequent tRNAs will enter the A site. Your second tRNA enters the A site. In this picture, our second codon is CCG, which if we were to go back and check the codon table, would code for proline, okay? And then what starts to happen, so you can see that we have 2 amino acids. It's important when you're building an peptide, polypeptide or protein, that what you need to do is take amino acids and link them by peptide bonds. So that's what happens during the third part of initiation. So I just want to kind of remind you what we have, EPA. Our first tRNA entered the P site. Our second tRNA enters the A site. And in order to start this process, you'll notice what happens then is that when we have 2 amino acids, this doesn't show the exit tunnel. The exit tunnel would be like right here. And you can see coming out of the exit tunnel, you have formation of that peptide bond. So you'll notice that this is not created on the amino acid that's like held in the P site, but technically this growing peptide strand is like above the P site. So I apologize for the weirdness between those two pictures, but you'll notice this is happening above the P site. But what happened, again, we have our first tRNA carrying methionine. Our next tRNA entered the A site, and you'll notice what happened is this amino acid is popped over and a peptide bond is created between these two. Just like that. All right, and now you have that second tRNA and it's carrying the growing polypeptide strand. And what we need to do is get this empty tRNA out of there and move this second tRNA over into the P site so that we can have another amino acid brought in. And that is elongation. So you can see what starts to happen is we have that second tRNA that moved over into the P site. It's carrying that amino acid strand. That very first tRNA pops out of the E site. This leaves the A site open. We can have that tRNA come in here, carrying our third amino acid. It keeps moving 5 to 3. And you can see this is what we saw in the picture before, right? So this one enters, and then that polypeptide strand is popped on over to that one. So we added an amino acid. We keep moving to the right. This keeps happening, et cetera, et cetera, et cetera, so on and so forth. So you can see what happens is we move all the way from the 5 to 3 prime strand, but eventually in the mRNA strand, you will have in the A site one of those stop codons. In this case, this is showing a UAG. So importantly, those stop codons do not code for tRNA. Instead, they code for a protein called release factor. And what happens is you get to a codon with release factor. That release factor basically just sort of like pops apart the entire complex, which you can see after you get release factor binding. It just sort of like knocks the whole thing apart. So the ribosome disassociates with the mRNA, tRNA, and then you have release of your polypeptide strand. All exciting. Okay, so I do want to look at this more closely, but first, I have a couple of questions for you. So let's try a poll, shall we? Okay. So our first question is number 5. So please, if you wouldn't mind, tell me which site the very first tRNA binds to. Remember, the very first tRNA is carrying methionine. What site of the ribosome does that very first tRNA bind to? So you have 30 seconds. Oh, does everybody not see the poll? I have the poll launched. So you can answer in the chat box. Good if you did. But if you don't see the poll, good job, everybody. Okay, I don't think my poll is working, so we'll go ahead and do chat. Everybody got it right. Good job. The answer is B, which is the P site. Good job. Okay, you're right. So the very first tRNA, where is it? That very first tRNA enters the ribosome into the P site. Good job. You guys are rock stars. Okay, let's have another question. And then after that very first one enters, all the subsequent tRNAs bind and enter what site? Go ahead and enter it in the chat box. Good job. Yeah. Right? Perfect. All of the subsequent tRNA enter the A site. So you can see here, if we look at that, right, our very first tRNA enters the P site. All subsequent tRNA enter that A site. All right. Good job. Okay. And you know what? I have one more for you. Okay. During translation, the mRNA is read in which direction? This is a very important question to me, very important for your lives as biologists. Go ahead and answer in the chat box. Oh, yes. I see those very quick answers, and I very much appreciate it. Good job. You are correct, and the answer is B. Right? mRNA is in the 5' to 3' direction, is read in the 5' to 3' direction. RNA is in the 5' to 3' direction. Don't ever forget it. Okay. So, good job. You guys are rock stars. Thank you. Okay. So let's go ahead and move forward. I feel like we have a good idea for translation. So just to summarize translation, it, just like transcription, has 3 steps: initiation, elongation, and termination. For initiation, what happens is you have your ribosome clamp onto the mRNA. You have that very first tRNA, which will always be carrying methionine and bind to the start codon AUG. That binds in the P site. And then all subsequent amino acid carrying tRNAs will bind in the A site. So that's what I'm talking about when I say AA tRNA. It's just amino acid carrying tRNAs. And I have here specifically, because that start codon codes for methionine, it's the methionine carrying tRNA that binds to the P site. And then all subsequent amino acid carrying tRNAs bind to the A site. And then during elongation, the ribosome moves forward. So again, let's make this more specific. It moves 5 to 3 in the 5' to 3' direction. So that second amino acid moves into the P site, and then you can have your third amino acid carrying tRNA bind to the A site all the way down until you reach a stop codon. And what happens when you reach that stop codon is you have release factor bind, not a tRNA, and then the complex is released. So I am going to, hopefully I can do this. Let me figure out if I can do this. I am going to open this up. I want to see if this is the video I'm thinking of. Is this? Let me see.
Subunits of the...
Oh no. Okay. Well, okay. This is a good video here, but I'm going to let you watch it on your own because I want to show you in a minute one of my very favorite videos, which reviews this entire process. Okay. So before we do that, I just want to show you this picture, which is a little bit insane, but it's good. I think everybody can still see this. But what this is showing is we're going from DNA to mRNA to protein. So this is just showing the entire process all together. So you can see here in red, you have your DNA strand. DNA is double-stranded, anti-parallel. So it's very important to know that you use the template strand, which is 3 to 5, in order to create a complementary strand of mRNA, which is in the 5 to 3' direction. You do this via complementary base pairs. So you see in our DNA, that's 3 to 5, you have a T. This means that in our complementary mRNA strand, we will have the complementary nucleotide, which is A. G, C. If you have an A, remember in DNA, the complement is T, but since we're building mRNA, if we have an A, the complementary nucleotide is U. So we go all the way from down, remember this doesn't show it, but this starts at the promoter, the beginning, and it goes all the way till we end the terminator, or the terminal region, and then we have a strand of mRNA. This also doesn't show, I don't think it's showing, I can't tell, but this also doesn't show very well the fact that you have both coding and non-coding regions. So what happens after you get this mRNA strand is it is actually shortened because you're splicing out those introns. It does show that you have capping and a tail added. So 5' cap, untranslated, 3' tail is also not translated. But then you'll notice that the very first codon is AUG, which codes for methionine, and then we go codon to codon to create our polypeptide. So I had a question. What are you looking for to tell what the complementary strand? Can you clarify that question a little bit for me? So for the complementary strand, what are you looking for exactly? If you don't mind clarifying that question. So basically, a complementary strand, all that means is that if you have one strand, oh, here it is. How do you know what the complementary strand is? Okay, perfect. Okay, how do you know what the complementary strand is? A complementary strand is basically just any strand that is created via complementary base pairs, base pairing. So this is completely relative to what you are talking about. So for example, because I refer to the 3 to 5' strand of DNA a lot because that's the one, the 3 to 5' is what you have to use to build a 5 to 3 because of the fact that complementary base pairing results in the creation of an anti-parallel strand. So we have to use a 3 to 5' template to create a 5 to 3' product. However, because of the fact that we use complementary base pairing to create that product, any product of complementary base pairing is going to be the complementary strand. So for example, let's just look at DNA. If I'm focusing on the 3 to 5' strand and I want to build a DNA strand, okay, so let's say I want to go from DNA to DNA. I have a 3 to 5' strand. I would have that sequence be TGA, CGG. The complement or the base pair that is complementary to T is A. Remember, you have T, A in DNA, and C and G. Those are your complements. So if I have one strand, its complement, if you have T, is going to be A. G, C, A, T, C, G. So in this case, I'm using a template that is 3 to 5. Its complementary DNA strand would be in the opposite direction, 5 to 3, and it would have the complementary nucleotide strand, right? So you can see this is called the coding strand. In this case, I just called it the complementary strand because that was the product if I was going to build DNA. In the case of transcription, we're taking DNA and creating mRNA. So our complementary product is going to be that mRNA strand. Again, if we're starting with a 3 to 5' strand of DNA, our product, our complementary product, is always going to be in the opposite direction, which is going to be 5 to 3, and it will always be the complementary nucleotides present in that product. So again, for DNA to RNA, this means if we have a T in our complementary product, we have A, we have a G, C, A, U, and it all has to do with those complementary base pairs. Does that help? Does that kind of make sense? So it's all just talking about the complementary. Perfect. Okay. All right. So that was an excellent question. All right. So that's just how this process takes place. And at the end result of this, you will have a polypeptide strand. Okay. So I won't go too much into this, but I think a long time ago, this is like one of my very first years when I was teaching this in anatomy and physiology, actually. I got done talking about protein synthesis. Somebody was like, I just have trouble understanding why this is important. I was like, wow, okay, so that's a big deal. The reason protein synthesis is so important is because you are made of your cells, right? So like the instructions for creating you are found in the DNA in your cells. So we have to somehow go from that DNA into like all of the stuff that makes us, right? That builds a person or builds an organism. So how do we go from just like nucleotides and DNA to just like large giant things, right? We have to build it. And in order to do that, we have DNA, we transcribe that into mRNA, and then we take that mRNA and convert that into protein. So the way I kind of think about that, like why are there so many steps involved? The way I kind of like to think about this is sort of like this. So my grandma was an immigrant from Germany, and when she came over, she didn't speak any English. She spoke only German. She immigrated into Chicago and opened up a restaurant outside of Chicago, which is still going, by the way. Sorry, this is my great-great-grandma. Opened up a restaurant outside of Chicago called the Forest Inn. My dad's name is Forrest, so the Forest Inn, if you ever are familiar with that area. That's my great-great-grandma's restaurant. And she brought with her, obviously, like a ton of recipes. These are like delicious recipes, but they're all in German. So I'm fortunate enough to have inherited these recipes, but I can tell you I can't read German. And someday maybe I'll have time to learn it. But the important thing here is I have these cookbooks. That's not a picture of it. That's like, I found it online. But I have these cookbooks and they're all handwritten in German. One of those recipes is not for chocolate chip cookies, but it's like for this amazing dessert. I can't remember what it's called. But what my grandma did is she copied over the recipe in German into English so that I could read it, right? So that's kind of like transcription. You have this language that you can't read directly into protein. It's in a different language, DNA. And what you have to do is convert it into a readable language, mRNA. But that's obviously like, I can't just eat that recipe in English. It's on paper, all right? You have to turn that recipe into the actual dessert. In this case, you have to turn the recipe into chocolate chip cookies into actual cookies. That requires products, right? That requires flour and chocolate. Just like turning RNA into protein requires amino acids, right? And then folding. And that's translation. Turning just that recipe into the physical manifestation, right? Making that recipe so that you can eat it. Another analogy, like why this is important, is because if you think about my grandmother's or my great-great-grandmother's recipes that are in German, let's say that you want to make one of those recipes. You're like, Dr. C, can I borrow your great-great-grandmother's cookbooks? Listen, I love you all. I care about you all. And you probably could read German. And you're probably a better cook than me. But I'm not going to just give you my great-great-grandmother's cookbooks, okay? What I would do, though, is I would take those cookbooks and I would copy them and give you a copy, right? A scanned copy. That's kind of why, like, with DNA is like, it is the instructions for building you. It is so precious and so important. And in your bodies, it's stored in a nucleus, which is an extremely protected area, right? I have these cookbooks in an extremely protected area. your cells are not just going to be like, hey, I need to make this protein. Here's your DNA, and pop it into cytoplasm, you know, to any ribosome that asks. It's going to say, this is DNA. It is very important. I will give you a copy of it. I can make 1,000,000 copies. That's mRNA, right? It's a copy of what you need in the DNA, but it won't be through translation. It won't be like ruining our DNA, our instructions. So that's my analogy for this, like why it's so important, why it's protected, why we go through all these steps. And what I like to show this, we have a couple more minutes, and hold on just a second. Oh gosh, sorry.
What?
So many pop-ups. Okay, what I would like to show you, can everybody see my screen? Can everybody see YouTube now? Okay, great. Okay, so I'm gonna pop this open. And if you can turn your volume on, hopefully you have volume on because I'm talking. But what I'm gonna show you is this video, and what this video is going to show you is the process of transcription and translation. And a really important thing I want you to take note of is this video is taking place in real time. And I'll describe why that matters here in a minute. So watch this video, and then I want to talk about what we just saw. All right.
What you are about to see is DNA's most extraordinary secret. The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only
So I just want to, for a second, just kind of point out what you just watched is transcription. And a question that I get a lot about these processes is, with all of the nucleotides, like where are they coming from? And same with translation, like where do the amino acids coming from? So the answer to that is, if you remember, like in eukaryotic organisms, you have the nucleus, which is isolated from the cytoplasm. And it is sort of like a gel-like consistency. But one of the things that is sort of like a solute in the solution of the nucleus is a lot of those free nucleotides. So those are readily available sort of in the nucleus as well.
difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
It's a bit like taking the information of.
I can't handle this guy. It's like mansplaining and it's not even good. Okay, past that. Okay, so what you just watched was transcription. So what they did is they took DNA and made a copy of DNA into RNA. So that really giant blue structure, the structure right here, is your RNA polymerase. Okay, so we're going to go ahead and move forward, and I'll stop again in a minute. All right.
That process is like making, it's like running on a.
Get out of here, dude. Okay.
When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of...
If you want to know if that's what it sounds like when your RNA goes out of your nucleus, the answer is, I don't know. I love the sound though.
choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome.
There's your ribosome, small
It
and large.
translates the genetic information in the RNA into a string of amino acids that will become a protein.
There's your tRNAs,
Special transfer molecules,
little
the
red
green
tips.
triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
I love that video so much, you guys. That is one of my very favorite videos of all time. So just before I go forward, I just want to talk to you for a second about what we just watched, because it's really, really cool. Oh, where did you go? Okay, so in that video, you just watched transcription and translation. So you watched transcription and then translation. You saw it snake out of the nucleus and everything. What I want to point out is something that's very important, and the fact that they kept reiterating that process that you just watched. The reason I love it so much is because that video takes place in real time. So what that means is that in the time it took us to watch that video, which without that guy like talking about whatever with computers, without that guy, it's about 4 minutes long. And in the time we watched that video, we watched start to finish transcription and translation of the protein, if you heard at the end, the protein hemoglobin. So if you've never heard of hemoglobin, hemoglobin is on, we might have talked about it, I can't remember, but hemoglobin is a protein that's on your red blood cells. And it's extremely important for carrying oxygen to your tissues and then taking carbon dioxide away from your tissues after they've done cellular respiration, basically. So hemoglobin is extremely important for getting oxygen to your tissues, which is necessary for your survival. So obviously, hemoglobin is an extremely important protein that you must have to survive. And we just watched in 4 minutes is all it took, which is a relatively short amount of time. 4 minutes is all it took to go from the DNA in your nucleus to the actual physical protein hemoglobin, which can be used to carry oxygen. So what it means that that took place in real time, making that, so it takes about 4 minutes for that entire process to take place. Every single one of you has to make hemoglobin. You rely on it for life. And what that means is when you watched that video, that same exact process at the exact same time was happening in your body. At the same time as you were watching it on that video, you made hemoglobin in the same amount of time. I love that video. I think that is just amazing. And I know right now we're going through like some tough stuff, but what I want you to do is just like, no, like if you are alive right now, which if I'm talking to you, you probably are. If you are alive right now, then you just made hemoglobin. You just made hemoglobin successfully in 4 minutes. So even though everything's crazy around us, you are still functioning and you are still doing really, really good. So good job making that hemoglobin. Give yourself a pat on the back because you're rocking it. All right. There's my pep talk for the day. Good job making hemoglobin. Okay. So just really quickly, let's move back to the PowerPoint. So there's my pitch for why protein synthesis is so important. What I would like you to do to practice this process is I will be asking you on future exams and quizzes to basically take a strand of DNA and turn it into a protein. So I think what we'll do, I'm going to leave it here. We're out of time anyway, but I'm going to leave it here for next week. And what I'd like to do when you come in on Tuesday is I'd like to see what protein this strand of DNA will create. So do that, right? Do that now or do that between now and Tuesday, whenever you want to do it. I have a DNA strand at the top of this slide. Remember what you do with transcription and translation. Transcription is taking a 3 to 5 prime strand of DNA, building a complementary strand of mRNA, and then mRNA has codons that code for amino acids. So use the codon table here to figure out what your protein strand would be. And that's what we'll pick up on Tuesday. We'll do that together. Okay, so with that, I'll kind of leave the PowerPoint open for if you do have any questions. But if you have any questions, go ahead and ask them. And if not, have a wonderful day. Okay. I'll hang out for a couple minutes just to make sure anybody has questions. But if you have questions, please ask them. I'm here. Thank you. Have a wonderful day. Oh yes, question. Do we know when part 2 scores on the exam will be? It'll take me a while to grade them. So I am working on it right now, but it'll take me a while to get through them all. Luckily, I have asked a TA for help, so hopefully that'll speed it up a little bit, but I'm working on it. You too. I miss you all so much. Have a good day.